In every cell of your body, DNA is packed tightly to fit inside the nucleus. But this packing is not random. It is organized into structures called nucleosomes. Nucleosomes can be thought of as spools that DNA wraps around like thread. These spools are made of proteins called histones. The DNA wrapped around them is less accessible, while the DNA between them is easier to reach. This arrangement helps cells decide which genes to use. For example, cells in your skin and muscles have the same DNA, but different genes are active because their nucleosome patterns are different. To study these patterns, scientists use a method called MNase-seq, short for micrococcal nuclease digestion with deep sequencing. First, nuclei are isolated from a tissue of interest. An enzyme called micrococcal nuclease, or MNase, acts as an endoexonuclease to cut exposed DNA in the open spaces between nucleosomes while leaving the wrapped DNA regions untouched. This process leaves behind fragments of DNA protected by nucleosomes, which are about 146 base pairs long. Smaller fragments can also appear, representing DNA bound by proteins like transcription factors. Once the DNA fragments are collected, scientists use next-generation techniques to sequence them. Sequencing reveals the order of nucleotides in these fragments. By aligning the reads to that species' reference genome, researchers can create a map of where nucleosomes are located. This can tell us which parts of the genome are accessible and likely to contain active genes. To recap, DNA is wrapped around histones, forming nucleosomes. During MNase-seq, the DNA between nucleosomes is cut, and the DNA wrapped around histones is sequenced. As such, MNase-seq can reveal how DNA packaging changes across conditions. MNase-seq helps us understand how cells regulate genes in normal conditions and how this regulation can break down. For instance, cancer cells often show unusual nucleosome patterns that allow these cells to grow uncontrollably. The data from MNase-seq can also be used to study differences between cell types, how cells respond to their environment, and how DNA packaging changes over the course of development. By revealing how DNA is packaged, MNase-seq gives scientists a window into how genes are controlled in health and disease. It is one of many tools used to understand gene regulation.